Right. Good day. And here we are, officially less than a day before exams now. I am terrified. So let's just get on with this. Let's just get on. Let me try and adjust this camera so it actually looks good. What we are doing today? What are we doing today? Well, I'm going to be revising very quickly. I want to go over translocation because I'll be completely honest, I've kind of forgotten a bit of that, so I think it would be good to go over that. I want to quickly go over the guard cells because I kind of forget part of that. And then what I'm going to do is look at a couple past paper questions, and then that's it, okay? I'm not going to try and learn anything that I haven't learned already. We're just going to go with the flow here, recap some stuff, and then just, take it nice and calm. I'm sweating to death, so if I go off camera for like a minute, it's because I'm, drinking some water. So, let's start then with the translocation. So what we have here is, I'm just going to draw this very crudely. I love that word, by the way, crudely. So, what we have is the xylem and the phloem. Now the phloem, I guess are called the sieve tube cells, or the sieve tube cells. Basically what they are is, unlike the xylem, which is just one continuous, band made up of dead cells, the phloem actually have companion cells, which provide all of the necessary, they have a lot of mitochondria, for example, for all the relevant processes, which we'll not get into. But anyways, so you have your companion cells, and then we have all the other cells, I think. For context, I'm doing this from my memory, and then I'm going to check to see how much I know. So, first of all, you have active loading. The gimmick here is the hydrogen ions from, I believe, the companion cells move out of the companion cells via active transport. And this creates a electrochemical gradient. Because there's a shit ton of hydrogen ions here, not a lot in here. So now that we've established this gradient, what's going to happen is via co-transport, the sucrose molecules and the hydrogen ions are going to move back into the cell using a symporter. So now what we have is we have a shit ton of hydrogen ions and a shit ton of sucrose in the companion cells. And from what I can recall, this, does it leave by diffusion, or it'll leave by facilitated diffusion, right? I think. It'll basically go into the phloem, from what I can recall. Again, this is just my memory. So step 1 is hydrogen ions move via active transport. Step 2 is they go back in via co-transport with the sucrose. Step 3 is the sucrose molecules go into the phloem. This will obviously create a water potential gradient, because what you have now is a low water potential. So water is going to move in via osmosis from an area of high to low water potential. And what that means is an increased volume. It's becoming more turgid. I think there's a special term for this, like turgid pressure, something. But in other words, the turgid pressure has increased. Because now the volume is increased, which means the pressure is increasing, I'm pretty sure. So now, in the cells which actually require the sucrose, okay, what's going to happen is they are just kind of minding their business using the sucrose molecules, which means that there is now a concentration gradient, a pressure gradient, because water is moving out of the phloem into the xylem because the sucrose is being used up. So what you have is you have a gradient. You have a, it's going to be a high pressure here, a low pressure here. It moves from an area of high to low at turgid pressure, and that's how you get your sucrose then going into the sink cells. Right. I know that I've definitely explained that wrong to some degree, but that's what I can remember. I know that the electrochemical gradient bit's wrong, but I don't recall what happens to the sucrose after it goes via a symporter. I forget what happens. I forget what happens. So, let me go quickly grab... I hope my MacBook's charged, because it would be a pain if it wasn't. And it's dead. Great. You guys are going to have to bear with me a sec here. So yeah, that's the one thing I wanted to quickly go over. We'll check now how much of that I got right. If I got most of it right, that's very reassuring. Again, in an exam, you won't have to go into that level of detail, even for a 6 marker. Most 6 markers, you literally get about a half of the marks in a 6 marker for just using buzzwords, like, concentration gradients, osmosis, pressure gradients, hydrostatic, just spam buzzwords. It genuinely works. It's annoying that it works, but it works. So what I'm going to do is we're going to go to Physics Math Tutor, because they actually have a whole section on past papers. Again, Physics Math Tutor, one of my favourite resources. So yeah, we've done surface area to volume ratio to death. We've done gas exchange to death. Digestion and absorption, we've done that to death. Mass transport, that's actually the entire mass transport topic. We probably should... Yeah, mass transport in animals, mass transport in plants. Let's answer some questions. And then if we come on to this, we'll do it. Give 2 ways in which... Oh, I hate this question. You guys, this question pisses me off, right? Because it's so easy, but I and everyone else in the class got this question at least partially wrong. Give 2 ways in which the structure of starch is similar to cellulose. You literally get a mark. You literally get a mark for saying they both contain hydrogen, carbon, and oxygen. It's just like, bruh, come on. Like, it's so obvious that no one will put it. 2 ways in which they're similar. They're both... How would I word this? Their monomers are both glucose. They are both polymers of glucose. Or their monomers are glucose. Either one, I think, would work. 2 ways in which they're different. Starch is alpha glucose, cellulose is beta glucose. Oh, another way they're similar, glycosidic bonds. They both have glycosidic bonds. The structure of starch. Is this asking for a literal structural difference? Because surely one would be starch is it heliceal? I don't know how you pronounce it. It's like a helix. Cellulose is straight and unbranched. So yeah, they're both made of monomers. They both contain glucose, contain glycosidic bonds. Hydrogen bonding within structure. So yeah, I put the fact that they're made out of the monomer glucose. That they contain glucose. Okay. Starch contains alpha glucose. Yeah. Heliceal. Yeah. Okay. So we got that right, but yeah, it's an annoying question. Okay, here. Suggest and explain one way in which sieve cells are adapted for mass transport. This would be the fact that they have companion cells which contain many mitochondria for for respiration. Actually, is it? No. See, it was trying to deceive us. There's another question about companion cells. So there you would say many mitochondria for respiration to provide ATP for transport. One way in which sieve cells are adapted, I don't know. I don't know. Why? How are they adapted? I'm struggling to think. Oh, okay, literally no organelles, so it's easy to flow. I mean, yeah, that's an obvious one, but I overlooked it, clearly. Okay, that makes sense, sir. So again, it's literally just like mostly a vacuole. Yeah. Organic compounds synthesized in the leaves of a plant can be transported to the plant's roots. So this is on about the whole mass flow hypothesis. One theory states that organic substances are pushed from high pressure to low pressure. How is the high pressure produced in the leaves? Okay, so this is going to be... What specifically is this asking? How the high pressure is formed? It's going to be H plus and sucrose move via co-transport into companion cells. This reduces water potential, so water moves by osmosis from xylem to phloem. I'm pretty sure that's right. Water potential becomes lower because sugar... Okay, so hold on. We need to say sucrose enters not the companion cells and not by co-transport. It's literally hydrogen and sucrose enter the phloem. To be honest, I didn't even know they did that, but okay. So yeah, hydrogen and sucrose enter a phloem. This reduces the water potential, so water enters the phloem by osmosis, which increases... see, I didn't actually mention the volume there, which was a silly mistake. So I would have only gotten a mark or two, but... Yeah, okay, that helps fix part of our thing. So yeah, they go through a symporter back into the companion cells, but then they move by, I'm going to guess, facilitated diffusion. Contrast the processes of facilitated diffusion and active transport. Okay. Facilitated, you got your channel and carrier proteins, which are involved. It's a passive process, which is used for ions and large molecules. Active transport also uses carrier proteins, and it also is for ions and large molecules, but it requires ATP. Is that going to be what this is wanting us to talk about? Let's have a look. I love how the website just crashes. Facilitated diffusion involves channel and carrier proteins, whereas active transport only involves carrier. Yep. Facilitated does not use ATP / is passive. Yep. Whereas active uses. Yep. Okay, we got that. Facilitated. Obviously. I talked about ions. I missed out on the most obvious one. Down a concentration gradient, up a concentration gradient. That right there is why you don't rush questions, everybody. I know it's for 20-degree heat, which is kind of messing me up here, but yeah, that's why you don't. A good question, which I found, like one of the loveliest exam questions I've ever found in my life, was explaining the whole gimmick of each transport mechanism. It literally wanted you, for the question, it was 6 marks, and it literally said, like, describe the ways of moving across a cell surface membrane. So you literally had to say, you got simple diffusion, which is movement of molecules down a concentration gradient, and they have to be, non-polar, lipid, soluble, and what's the other one? Small lipid soluble non-polar molecules down a concentration gradient, and it's passive. You got facilitated diffusion, which is for ions / large molecules down a concentration gradient. It's passive, but it requires channel or carrier proteins. You got active transport, which is movement of ions against a concentration gradient using ATP via channel proteins, carrier proteins, osmosis, which is the movement of water molecules down a concentration gradient, down a water potential gradient from an area of high to low water potential. Is it water potential? Yeah. And then co-transport, which is movement of 2 molecules. It's the movement of 2 molecules. It's an active process, and it often involves a symporter. Honestly, guys, I'm knackered. Right, it's so warm. I'm just losing the plot here. So leaf cells actively Okay, hold on. This is an explanation. Leaf sugars actively transported into phloem. Okay, so it's by active transport. Okay. So the process goes: active transport of hydrogen ions out of companion cells. Yeah. Leaf sugars actively transported into phloem by companion cells, lowers water potential in the sieve cell, so water enters by osmosis, increase water pressure, causes mass movement. Okay, so the leaf sugars get actively transported into the phloem by companion cells. Yep, got that. That lowers the water potential, so water enters the phloem by osmosis. This increases the pressure, which causes a mass movement towards the sink. That's what it's called, mass movement. Let's go very quickly. I actually made a tiny little document with some general with some mistakes which I always make. Literally just a compilation of mistakes that I always make in exams. So let me just go on that quickly. My note document is not updated in literally a month. Hello? Update, please. Why is it not updating? Why is it not? Okay, I'm sorry, guys. This is. Um. Okay, well, I literally. I don't know why this is happening. Why it's not letting me access my notes. They just aren't syncing. So unfortunately, I'm not going to be able to do that. Well, before we carry on with some exam questions, I actually found a graph made by someone who's really cool. It's a very helpful guy. And this graph was actually very, very helpful. It basically showed that some of the most common topics that they want you to talk about in exams is mass transport, transport across membranes, infection and response, and proteins. So these are actually the topics which appear most in the exams. Mass transport, transport across membranes, infection and response, and proteins. So if you guys are genuinely struggling, right, if you have no idea, about the tracheoles on an insect and how they're, I don't know, thin to decrease the diffusion distance, shorter diffusion pathway. I don't know what you're revising. But look, if you're genuinely struggling, just focus on at least two of these topics. For me, I love infection and response. It's like my favourite topic. I know a lot about it. And I would argue the protein topic is also very nice, when talking about enzyme collisions, the higher the temperature, the higher kinetic energy, more collisions. I could write about that for days. So these two topics right here are lovely. In my opinion, transport across membranes, that's a pretty easy topic too. I think active transport, co-transport, the only thing which people don't like is co-transport. I think all the other four are easy as. Even mass transport with the heart, it's honestly really easy if you kind of take time to think about it, right? So you got, the way I draw it is one going in, one going out. That's how I kind of represent the heart, right? And it's a kind of very crude drawing, but you know, you got the vena cava going in, the aorta going out. You got the pulmonary artery, the pulmonary vein going in, and that's basically all there is to the heart. You got your pacemaker cells here. You got your thicker muscle tissue here. Um, you know, you got your three processes: cardiac diastole, um, atrial systole, and ventricular systole, and that's literally all there is to the heart, really. Even things like I mean, quick overview of the structures. Quick overview of the structures. We got the veins. So these have I believe, anyways, a wider lumen. They are thinner, less muscle tissue. But on the other hand, you have arteries, which have a narrower lumen, thicker muscle and elastic tissue. Veins have valves on them to prevent backflow because they're going into the heart. Arteries are able to relax, and this can increase blood pressure. This increases blood pressure to ensure that, you know, they're able to deliver their blood, their oxygenated blood. Capillaries, one cell thick, they have pores to allow for, you know, tissue fluids. That's all there really is to it. And then you have the arterioles, which then branch off obviously into the capillaries, and then the venules, which, you know. But arterioles obviously have the thick muscle tissue and they're able to relax. So let's go very quickly, before I pass out from heat exhaustion, let's just go look one last time. Oh. Let's just look one last time at some generic exam questions. What we'll do... Yeah, like, the mass transport topic is arguably the biggest. But look, you got hemoglobin with the, you know, Bohr effect. Each ion can carry one oxygen molecule. That's easy stuff. You know, loading, unloading, affinity, dissociation, association, Bohr effect with carbon dioxide, circulatory system, capillaries, one cell thick with tissue fluid, valves, arteries, muscle layer, thick walls, high pressure, phloem, the mass flow theory, active transport of sugars, sieve tubes. Oh, ringing and tracer experiments. Ringing is where you cut off the outer ring of, I guess it would be like a branch of like a plant where the phloem is located. So you're removing the phloem, and that will basically show that because the sugars can no longer reach downwards, you have new, I guess, leaves growing underneath where the ring is. And the tracer experiment is just using a radioactive carbon isotope that's then put in the CO2 so that when it does photosynthesis, that CO2 is then passed around the whole thing. Xylem with a cohesion tension theory. Basically, as water from the bottom is pulled up, oh my god, my head is killing. Basically, what happens is in the leaves, water is used for photosynthesis. That causes there to be a water potential gradient. So, let me just quickly look. Let me just quickly check that. See, the heat is getting to me. I don't know left from right. Let's have a look. Yeah, the evaporation of water due to the hydrogen bonds, cohesion tension. So honestly, you guys, from this whole thing, the thing you need to know. So yeah, elongated tubes, they have, what's it called, lignin, to prevent adhesion. Okay, so number one, the water evaporates via transpiration. This leaves the cells with a negative. Okay, so yeah, it lowers the water potential, so water moves in via osmosis. The water tension pulls the water into the leaf, and as a result, due to the fact that they're cohesive, this then pulls the entire column of water upwards, and then more water enters. Okay, so yeah, I'm gonna. Oh, my leg. I'm gonna wrap things up because look, let's be honest, right? At this point, there's no point in trying to learn anything new if, it's flipping 22 degrees outside and I'm struggling to put a sentence together. So what I'm going to do before we wrap up and before I wish you guys good luck and, tomorrow I'll do a stream after it's done letting you know what I thought. Before we go, I'm going to make a quick table. Alright, I'm going to make a quick chart. This will span the entire whiteboard. And what this will be is this is going to be topics which I hope come up, topics I hope do not come up. And you guys can let me know what you want to come up, what you don't. So what I want versus what I don't want to come up. And I'll quickly go through some of them too. Okay, number one. Monomers and polymers as a whole is a great topic. Monomers and polymers, but the whole biological molecules topic is lovely. So anything to do with, what's the definition of a monomer? What's the definition of a polymer? That would be lovely. I think, carbohydrates. There's nothing in carbohydrates which I struggle with. I think, honestly, the most difficult ones will be those obvious questions where they want you to, reach a little bit further. Lipids, I think, would be nice. I've recently gone over, the whole lipid topic again to revise because I was kind of struggling. I think if we get a question where it suggests why and it's to do with lipids, I think I might understand that better now. Remember, it's a polar head, a non-polar tail, and that's why it's able to form a bilayer. Proteins and enzymes, lovely power. I think now I know about the whole kinetic energy theory, I think I can write about that for days. ATP is a pretty nice topic. Phosphorylation, basically, I'm pretty sure anyway, it increases the reactivity. It allows a substance to become more reactive, or it changes the shape of a substance. That's what phosphorylation is. And then the energy used can be used for, active processes like active transport. Let's see. DNA, I think, is nice. DNA replication, the whole semi-conservative thing. Maybe we get a question about semi-conservative, which would kind of suck. But remember, it's Watson and Crick. Those are the two fellas that came up with it. Even RNA, the whole transcription, translation thing, I'm calm with. I think I've really understood the definitions. In an exam, I feel like I would get about 3/4 of the marks for a question about it. What else? tRNA. tRNA with an active, the whole thing of basically it binds to a codon. The complementary. Oh my god, bro! I can't think. I better not be like this tomorrow. The active. Oh my god, bro! What am I saying? The anticodon, the complementary anticodon, binds to the codon, and then tRNA brings an amino acid to join the polypeptide chain, and then the polypeptide bonds form, and yeah. What else? What else would I be, like, I would be gassed if it came up. Anything to do with microscopes? Anything about antibodies? Bro, I'm gonna be so happy if we get a question on phagocytes. Monoclonal antibodies. That's just about testing, really. The ELISA test, I think would be nice. The whole thing could be stretched to a 5 or 4 mark, which would be lovely. You know, talking about, you know, in your, what's it called? Not the container. In the well, you have antigens. Yeah, you have HIV antigens. The HIV antigens, you then put in the blood, and if a person does have HIV, then their antibodies will bind to the antigens because they have, you know, the same shape, they're complementary, whatever. Obviously, then you will wash it out. Bro, I'm burning. I can't do this anymore. What else? What else? Very quickly. I think I'm calm with insects and fish. The whole thing of them, you know, breathing. I think that's fine. But mitosis, lovely if it comes up. What I don't want to come up, number one, meiosis. That's going to be a vile topic if it comes up. I think anything, literally any ecology related would suck, to be honest. Anything ecology related, you know, describe biodiversity. I can do the calculations just fine, I can interpret things, but I cannot tell you the definition of biodiversity or species richness. Or I know what the index is. That's the... And I forgot what it is. So it's the relationship between the total number of organisms in a habitat and the number of organisms of each species in a habitat. I think the practical stuff, things where we have to talk about practicals, that's like 50-50 right there. Because if we get asked, you know, a generic question, it could be nice. If we get asked a really awful question, it will not be nice. You know, things like, oh, what happens if you push down, why would you push down on the microscope slide? Well, that's easy. Because then it makes it thinner, makes the sample thinner, so that light can pass through. But if you get asked, you know, a weird question, it might not be. Protein synthesis, I've already said, would be pretty nice. Lipid digestion. I'm actually quite good with lipid digestion, but I think in an exam situation, I don't know whether I'd be able to recall every little detail. I think carb digestion and protein digestion is lovely. I think those two are lovely if they come up. But can you guys see? But bear in mind, like, I'm just a regular student, right? You know, I revise like maybe over the course of a week. I'll revise like 3 or 4 hours at home. You know, not too much, I would say. But despite this, see how many topics are overwhelmingly good. If they came up in an exam, I would be happy. Versus how many topics I don't want to come up. You know, it's like, there's like 20 here, there's 3 here. So like that really goes to show, you know, with just literally a couple of hours of revision a week, you know, originally, I'll be honest, I think this topic that topic, that topic, that topic, that topic, all of this, that topic, and I think all maybe even microscopes. All of us with an orange dot. I would have panicked if they came up in an exam a couple of months ago. But because I've clocked in and I've started revising, doing these streams, doing past papers, I think now most of these I'd be happy with. Let me just go quickly look through. This is a past paper which I did a while ago. Describe how a complementary strand of DNA is made. So your free-floating DNA nucleotides will, is it join or bind? Or will pair to their complementary nucleotides. Phosphodiester bonds are formed, obviously. Yeah, I could definitely word this better. Your free-floating DNA nucleotides pair to their complementary nucleotides in the DNA strand. Phosphodiester bonds join the adjacent nucleotides together because of the action of polymerase. Yeah. Contrast the structures of DNA and mRNA. DNA has introns. RNA has no introns due to splicing. DNA is a double-stranded. RNA is single-stranded. In RNA, thymine is substituted with uracil, but DNA does not have uracil. It has thymine. There you go. DNA has thymine, but mRNA has uracil. Yeah, that would be a better way of wording it. Describe how you would test for the presence of a lipid sample. Add ethanol, add water, shake, and it's positive if you get a milky white emulsion. Describe how a saturated fatty acid is different from an unsaturated. Saturated have the double bonds. They don't have the double bonds. Unsaturated have double bonds. So saturated have the maximum amount of hydrogen atoms abound. That's the way you're meant to say it, I guess. Why can't lipase digest this random ass substitute? Because the active site is not complementary, and so it's unable to bind. The substrate is unable to bind to the active site. Why can't this cross the cell surface membrane? Because it's too large of a molecule. 2 ways in which hydrolysis of ATP is used in cells. Number 1, 2 ways in which, provides energy for active processes and phosphorylation of other substances. I'm just going through the answers, by the way. I'm just kind of recapping, making sure I know. Oh, name a part of a mitochondria labelled X. It was pointing towards the foldings in the membrane, which are called the crista. And they provide a large surface area. Do we need to go through the structure of a chloroplast quickly? Because, hold on. No, I think I've got it. What evidence is there that it's a phospholipid boiler? That's a big question. The graph question. Oh, this is a good one to remember. You have to plot the proper. You have to plot everything. I would have gotten 0 marks because in this question it's on about what can you conclude. You would have had to talk about, how one fact is linked to another. The induced fit model, this is where the active site is not initially complementary. The active site is not initially complementary, but the shape of the active site slightly changes to be complementary to the substrate. Oh, this is a weird one. Tangent lines. Didn't know you had to do them anymore, but you do. Explain the results. I rambled, but it was asking you to say the substrates are used up, so the rate slows down. This one was on about colorimeter. I can't remember what this wanted you to talk about, but basically you just need to say something like, measure the absorbance, plot a calibration curve, and use, the X or Y axis. Would it be better to give 2 or 3 doses? You should give 2 because there's more antibodies. There's a similar amount, so it would be more expensive to give 3 anyways. The t-test allows you to compare means. Two ways you can use base sequences to compare different types. This is literally in the question. You compare base sequences of DNA, compare base sequences of mRNA. Chromosomes are not separated. There are double chromosomes in one cell, so one cell has 0 chromosomes. Right, well, I'm basically done. Right, I'm done. I am absolutely beyond knackered, so I'm, for the rest of the day, just gonna chill. I'm not doing any more revision, so that's all for today. If you guys, if you guys are worried for this exam, try not to be. I know that's bold coming from me, because I always panic when it comes to exams. But, again, as I was showing people yesterday, it's honestly not that bad. If you want to get an A in this exam, which I'm sure all of us want to, the least you have to get is about 40. If you get 40 marks per paper, then you've got an 80 out of 75. That's 80 out of 150, sorry. That's above 50%, which means you're likely to get an A. If you really want to guarantee an A, you have to shoot for 45 marks per paper, or 90 total. So if you ace one paper, let's say, what I always do in the exams is I try and count how many marks I think I've gotten, and then I go to questions where I think I've gotten no marks and try and cram in a load more detail, or, rethink. So a very big thing is if you count through, for example, paper 1 tomorrow, and you see, oh, I think I've gotten, I think I've dropped 20 marks overall, 55 out of 75. Then well done, because that means you only need 35 marks in the next paper. You need 20 less marks. So the more marks you get in this paper, the less marks you have to get in the next one. And the next one is usually the worst paper. That's where you get ecology questions, meiosis questions. That's where typically you get less reliable things. So try and ace paper 1, because then you will ace paper 2 just as well. That's my advice anyways, that's my advice. Again, none of us know what it's going to be until we open the paper. None of us know. I mean, look, some more reassuring things. The GCSE paper for biology paper 1 was nice, apparently. There were a lot of 6 markers, but it was nice overall. So maybe they'll nice us. Who knows? Good luck, everybody. And yeah, stay happy, healthy, and safe. Good luck, everyone.